Hi, my name is Malvina Marku and I am a postdoctoral researcher in Vera Pacali's lab in Cancer Research Center of Toulouse in France. In this presentation, I will present some of our recent results in data-driven gene regulatory network inference of chronic lymphocytic leukemia cells. The work presented here is part of our broader aim in our lab, which is to study the tumor microenvironment. As we know, the tumor microenvironment is a complex and dynamic ensemble of cells, including, of course, cancer cells, various immune cells, fibroblasts, blood vessels, and multiple chemicals like cytokines, microRNAs, growth factors that establish the interactions between the cells. Importantly, several studies have shown that certain properties of the tumor microenvironment, like its cellular composition, can play an important role in disease progression and response to treatment. Particularly of interest, monocyte-derived macrophages are one of the most abundant immune cells in tumor microenvironment. They are highly plastic cells and cover a wide spectrum of polarization phenotypes, but however, they are generally classified as pro-inflammatory, or so-called M1, or anti-inflammatory, so-called M2. In the tumor context, we refer to these phenotypes as antitumoral and protumoral, respectively. In this context, a distinct phenotype is identified, TAMS, for tumor-associated macrophages. In our lab, we are working with chronic lymphocytic leukemia, which is a blood cancer associated with the presence of highly proliferative malignant B cells, which proliferate in the lymph nodes and circulate in the blood. Importantly, when in contact with macrophages, they are able to educate them and polarize toward tumor-associated macrophage phenotype called nurse-like cells in this cancer. The complete molecular mechanism of this interaction and how it can lead to resistance to therapy is yet to be understood. To help tackling this question, we have performed in vitro experiments where we co-culture the monocytes purified from healthy donors with CLL cells from cancer patients. RNA extraction from both CLL cells and macrophages was performed every day for a time window of 13 days, resulting in a dataset of RNA-seq time series of a total of 2 patients and 2 replicates each. Here we are showing the result for the CLL cells only. The broad aim of this study is to build a multi-scale model of CLL NLC crosstalk. Our lab has previously built a gene regulatory network of macrophage polarization and applied a logic dynamical model to identify the main polarization phenotype of macrophages in the presence of various extracellular signals. With the RNA-seq time series, we aim to build a data-driven gene regulatory network of CLL cells in order to infer which molecular pathways are involved in this crosstalk. Inferring gene regulatory networks from an RNA-seq time series has been an evolving computational topic for many years. With the dataset that we have collected, we used DGNI3, which is an inference gene regulatory network inference method based on ordinary differential equation and random forest algorithm. To reduce the complexity of the inferred network and its size, we focused only on the transcription factors in the time series, and the resulting network is a directed and weighted network. After the inference, we performed topology analysis and G-set enrichment analysis. The inferred regulatory network of CLL cells look like this in this figure in the left. Here we applied several topology measures like pitch rank, burst constraint, alpha centrality, etc., in order to identify the key regulators in this network. The analysis identified transcription factors like the stop TFs proven to be involved in various processes in CLL cells. Then, G-set enrichment analysis was performed to identify the biological processes the transcription factor in the inferred network are involved in. The analysis showed enriched processes like cytokine production regulation or rhythmic processes recently proven to play an important role in cancer cell behavior. Particularly in cytokine production regulation, we were interested in these specific cytokines produced when CLL cells come in contact with NLCs. To check how the TFs in our network are related to the production of the cytokines, we used Reactome functional network, and we observed that indeed The inferred TFs are involved in the regulation of the cytokines in the crosstalk between NLC and CLL cells. To conclude, by performing gene regulatory network inference from RNA sequence series, we were able to identify important transcription factors involved in specific and important biological processes in CLL macrophage crosstalk, like these two specific cytokines, which are proven to drive the macrophage polarization toward a pro-tumoral phenotype of macrophages, which is the NLC phenotype. Additionally, topology analysis on the inferred network identified determinant TFs involved in various processes in CLL cells. In perspective, we aim to develop a dynamical model of CLL cells and integrate the molecular models of both macrophage and CLL cells in an integrated model in order to study how the system behaves both by considering the cellular interaction and the internal molecular regulation. I conclude by giving an overview of our lab main research line and expertise, and I thank you for your interest.